And this morning's plenary speaker will be Peter Macreadie. Peter has been working on seagrasses for over 10 years. He started off working in biogeochemistry, has done a series of work on fish assemblages associated to seagrass, and has more recently moved to Keeling University in Southern Australia, where he's leading the blue carbon lab, and that's the focus of his current research and the focus of his talk this morning. So welcome to Peter.
Well, thanks for the introduction, Rich. It's a great honour at this stage of your career to be able to give a plenary. So I wanted to know, have you ever felt so deeply and hopelessly in love with somebody or something that you thought you couldn't possibly love them anymore? Like you had maxed out, you couldn't love them anymore, and then they go and do something that makes you just somehow find more love. And for me, this was what happened with seagrasses. I've been studying seagrasses. I love them for their ability to cycle nutrients, support fisheries, valuable nutrition for hundreds of millions of people. And then I learned that they're locking up 20 billion tonnes of carbon, and by doing so helping regulate our planet's thermostat. And then on my journey here on the plane, I sat next to my good buddy and close colleague. I said I wouldn't mention his name, but it rhymes with mauled pork. And he said to me, when we were reflecting on that I'm giving a plenary and who might get fired for this, he said, I'm sick of hearing about blue carbon, it's just mud. And I gave a little chuckle, but I was crying on the inside. And I thought, well, you know, as a scientist, you've got to take these comments on board and you've got to reflect on it. So I've updated my title slide to Seagrass Blue Carbon, Is It Just Mud? So first I want to talk about climate change. And we hear so much about the need to switch to green technologies, to reduce our reliance on fossil fuels, to reduce the amount of carbon that we release into the atmosphere, which is accelerating climate change. But there's another aspect of dealing with climate change that we don't hear about so much, which is what do we do with all the carbon we've already released into the atmosphere? So carbon that will be floating up there for hundreds and hundreds of years. And even if we stop emitting carbon tomorrow, we will still face a 1.5 degree increase in global temperature. And so the answer is, or an aspect of climate change we don't hear so much about, is biosequestration. So biosequestration is the process of removing atmospheric emissions and putting them back in the ground. Biosequestration is when plants and other photosynthetic organisms take up CO2, they breathe it in, they keep the seed, that's the carbon, they release the oxygen. That carbon can be put into the ground, it can be bound within plant material, which forms organic matter, which goes on to form our rich carbon stores, which is what created our fossil fuels in the first place. So it's an aspect of climate change, part of a two-pronged approach we don't hear so much about. And we have reason to believe that biosequestration is a really big deal. If you look at the CO2 curve that's represented in the Keeling curve, this is the longest running record we have in history of CO2 emissions through time. We see that CO2 emissions are rising, and with it we know temperatures are rising. But if you look closely at this red line, you see it's actually oscillating. It's going up and it's going down. The CO2 levels are going up and down. When it goes up, that's when our northern hemisphere plants are respiring and releasing CO2, and when it's going down, they're absorbing carbon and they're reducing atmospheric CO2 levels. So we know that biosequestration is a big deal, and not surprisingly, we think about our trees. But we can't rely on our trees. The trees are very important. The amount of land area we would need globally to really offset and put a dent in our emissions with trees would be unrealistically large. We also know that the carbon is bound within trees for 50, maybe 100 years for the lifetime of a tree. And when they die and break down, they release that carbon dioxide back into the atmosphere. Some of that carbon is stored within soils, but there's relatively high turnover, and they tend to reach saturation points. The other thing about forests is that they sometimes catch on fire. And so there's been this quest to try and find other ecosystems that are important for biosequestration. Other habitats that can lock up carbon for longer periods of time, systems that don't catch on fire. And so for us, for me, I joined this quest largely as a result of this document. And I remember the day it came out. I was sitting in a meeting, I just printed it off, it was a thick wad of a document, and I was flicking through it thinking, wow, these numbers are right. This is huge. For the seagrass world, in terms of conservation initiatives, blue carbon, these numbers stack up, this will be fantastic. So in the summer of, must have been 2009, while most of Europe was partying, Carlos Duarte was writing most of this section on blue carbon, and it's had a huge impact, and it was a great report. Interestingly, back in 1981, which was the year of my birth, we also saw a paper come out in Science, also saying that marine macrophytes are really important global carbon sinks, but the timing just wasn't right. And the way that it was pitched in this document just had so much impact. So I remember when I read this document from that day forward, I thought, okay, I've got to get onto this blue carbon thing. This is going to be a great opportunity for seagrass conservation. So blue carbon, it's carbon that's sequestered and stored by the oceans. But as Rod Connolly noted when he gave his blue carbon plenary in China, the heavy lifting is really being done by just 3 habitats: the seagrasses, the tidal marshes, and the mangroves. These 3 habitats only occupy 0.2% of the sea floor, yet they sequester more than half the ocean's carbon. So, one of the things that has been very impressive about blue carbon, and here is data from McLeod's paper in 2011. What I've done is, you might recognise this, but I've removed the log scale. I think it does a bit more justice when you remove the log scale. It shows that blue carbon ecosystems sequester carbon, capture and store carbon in soils 40 times faster than tropical rainforests like the Amazon. And so what that means is for every 1 hectare of blue carbon habitat, you need 40 hectares of green carbon habitat to do the same amount of carbon offsetting. We also learnt that they represent very long-term carbon sinks. They can lock up the carbon in the ground for thousands of years. And also, they provide many valuable co-benefits, including they support dugongs and turtles. Now, you know all this stuff, and I woke up at about 3 this morning. I'm still in a different time zone, and as you do, I started thinking about seagrasses. As I'm there, tossing and turning, thinking about seagrasses and thinking about Carlos's plenary last night, and he was saying, seagrasses are the ugly ducklings of the marine world. And I had it was about just before 4 a.m. I had an epiphany, and I know that what I'm about to say is probably going to offend most people, and I pulled out the codes of conduct this morning just to check. I think it was just the Australians. When the Australians walked into the registration, we were all issued with a code of conduct, and I think this must have been something to do with what happened on the post night in Brazil, which was regrettable, and we're all very sorry for it. And Joel Creed, we've apologised, but in China we were very well behaved, and this so far we've been very good. But I've looked at the code of conduct, and what I'm about to say, even though it might cause offence, I think is nothing but the truth, which is that yes, we have an ugly duckling, but we also have a very ugly baby. So seagrasses, to us, look amazing, look beautiful, but to the rest of the world, they don't. And if you look at this picture and you're thinking, what's that sea pig doing in the way of a beautiful shot of seagrasses, you might know what I mean. Yeah. Or if you've been down to the coast and your friends are all looking at the coral, or the fish, or the rays, and you're in a little puddle saying, woohoo, there's 5 species in 1 metre squared, and you're getting excited about that. I think this is something that we're starting to realise, is that we don't see seagrasses the way everybody else does. I mean, I think they're gorgeous, but just wanted to put that out there, that we might have an ugly baby on our hands. So I think these charismatic megafauna can be really helpful for the cause. We know also that our seagrasses support half the world's fisheries, valuable nutrition for hundreds of millions of people. Many of these people are from developing nations. Many people are living hand to mouth and rely indirectly on these seagrass meadows. We also know that seagrass meadows stabilise our shorelines and prevent coastal erosion, reducing our need for grey infrastructure, which is particularly important with sea level rise and increasing storminess in the coastal zone. We know that they have important connections with other ecosystems like the mangroves and the coral reefs. Macreadie received an ARC, Australian Research Council, linkage grant this year to study deep water seagrasses around the Great Barrier Reef. Back in the mid-80s, Rob Coles had discovered there's 40,000 square kilometres of this stuff around the Great Barrier Reef. Who knew? And so, what are these deep water seagrasses doing in terms of supporting our corals? Are they helping buffer against ocean acidification? Are they providing an important nursery habitat for fish on the Great Barrier Reef? So, the research that my group does is really around trying to build a business case for seagrass restoration, conservation. And even though I felt quite dirty about doing this initially, we're putting dollar values on the seagrasses because that seems to be quite effective. And what we're trying to achieve is getting ourselves in a position to say, Mr. Prime Minister, here we have an innovative solution for helping to mitigate climate change, for contributing jobs, improving natural capital, improving community well-being, and to be more specific in the seagrass space, we want to be able to say, you know, for every $1 million we invest in seagrass restoration and conservation, we're going to get $10 million back in terms of benefits to our blue economy. Or for every 5 hectares of seagrass we restore, we're going to offset the emissions of 500 Australians. These are the sort of numbers that we're working towards coming up with. And so as part of this research with our colleagues and building a business case, there's 3 steps. The first is looking at the distribution of blue carbon. So where is blue carbon? I remember the first job we got when we moved to Deakin University, the first big job we got, the Catchment Management Authority said, have we got blue carbon? How much is in our backyard here? We've got seagrasses, we've got salt marshes, we've got mangroves. So, we took up the process of coring those environments just to show how much carbon there was. And then valuing that blue carbon, monetizing it. It's amazing when you monetize something, how you can demonstrate worth and value. In the seagrass world, because the price on carbon is so low, it doesn't really represent their true value. At the moment, we've got carbon, which is one of the biggest threats to humanity, carbon in our atmosphere, and we price it in Australia, it's only $12 a tonne, it's pretty cheap. We've also been understanding the dynamics of blue carbon. So if we have environmental changes, if temperatures go up, if we lose water clarity, what is going to be the response of seagrasses and their carbon sequestration capacity? And with that information, we put that together and we're developing decision tools to help predict how seagrass carbon sequestration capacity will change in the future. So we know what we had in the past, what we have now, and what we could have in the future under different management scenarios. And finally, and this is probably where it gets really tough, how do we establish new seagrass carbon offset initiatives? How do we develop a market for blue carbon? How do we put policies in place to help conserve seagrass meadows based on their blue carbon sequestration capacity and also these other ecosystem services as well? So this is just a little video of what a typical coring campaign would look like for us. We're working in a salt marsh here, a tidal marsh here. Banging in a piece of PVC. This is about as simple as it gets. Banging in a piece of PVC poling, taking out some mud, some soil, and we take that back to the lab, we section it and we measure the amount of carbon and we come up with a value for the carbon stock. This is a short core, but typically we go down to about 1 metre. This is an area of the southeast Australian coast where we've sampled 200 wetlands, seagrasses, tidal marshes, mangroves, but also inland wetlands as well, working out the carbon stocks and the accretion rates. And one of the most important things for us is using that information, generating what we call a blue carbon heat map. This blue carbon heat map shows an individual estuary and we can see where are the blue carbon hotspots, where are the high levels of carbon. And resource managers can instantly say, oh, that's good to know, we were planning on putting a marina there and that's where the highest levels of blue carbon are. We'll shift that marina or we won't go ahead with that development. So we found mapping to be a really useful tool with communicating with our resource managers where the blue carbon stocks are now and where will they be in the future. So how much seagrass do we have globally? Well, Jim Forker and colleagues have done the hard work for us. Back in 2012, they published a paper in Nature Geosciences, which estimated that seagrasses globally store 19.9 petagrams of organic carbon. And unless you're in the carbon world, you're probably thinking, what the heck is a petagram? So to visualize this, here is 1 tonne of CO2. That's what it would look like. Seagrass meadows lock up 19.9 billion of these. That's how much is bound, or estimated to be bound in the top 1 metre. But even that's still hard to visualise. 19.9 billion, what does that look like? So I've grabbed what I think is a very useful video from a group called Climate Visuals. And what they show is real-time release emissions from New York City, which is 54 million tonnes of carbon per year. And this video basically starts the clock and shows what it would look like in a day, in a month, and in a year with these 1 tonne bubbles of carbon. So if you're wondering what you produce as an individual in terms of your annual emissions, the average UK citizen produces 7 of these bubbles per year. The average US and Australian citizen produces 18 of these bubbles. Somebody from Somalia, 0.1. My flight here alone was 3.2 of these bubbles. So that's a lot of carbon through flights. Eating meat will set you back 0.8 tonnes of carbon per annum. If you, and I didn't come up with this one, I read this one. If you make love 2 hours a week for a year, you will save, and you turn off the light, you will save 0.9 tonnes of carbon per annum. But if you have a baby, it'll set you back 10,000 tonnes. So this here shows CO2 production, and in a moment there, it'll show you what this build-up looks like after a day. And we're releasing from damaged seagrass meadows 299 million tonnes per annum. This is 1 hour of emissions. Can I see that okay? And next we've got 1 day's worth of emissions. It's just from the city of New York. And then this is 1 year's emissions, 54 million tonnes. And seagrasses currently lock up 19.9 billion. So we're going to hear some great blue carbon talks in the next session that are really going to go into the dynamics of blue carbon sequestration. And I just wanted to pull out some of the highlights we've had over the last 4 or 5 years. The first one is that we've learnt that managing for blue carbon requires catchment level management. We can't be focusing just on the meadow. We need to be understanding what's happening upstream. And that's because geomorphology is a major driver and influencer of carbon sequestration by seagrass meadows. There's a high level of land-sea connectivity. So things that are happening high up in the catchment, whether it be erosion or dams or forest fires, these processes can influence the amount of carbon that washes down into seagrass meadows. Hilary Kennedy published a paper with colleagues showing that seagrass meadows, about half the carbon they lock up, they never produced. It was external carbon, allochthonous carbon, that they sequester and they trap and store, and they're very efficient at it. I mean, they create these anoxic environments, they trap what runs off the land and through the water column, and they bury that very efficiently, and they minimise the amount of microbial activity that would break down that carbon. Oscar Serrano and I wanted to know, could mud be a cheap indicator of blue carbon content? After spending tens of thousands of dollars analysing carbon samples, we thought, maybe mud could be a good indicator, and I think so my colleague might have been right about the role of mud. And what we found is that the answer is yes, sometimes. So sometimes mud can be a good indicator, but it tends to be only environments where there's a large portion of external input, allochthonous inputs, areas where Halophila grows, Haliduli, these fast-growing species, Zostera, where they might not be making a lot of carbon themselves. And so we reviewed the data over 2,000, a bit over 1,000 samples, and saw that in many situations we do find a strong relationship between the amount of mud and the amount of carbon, but in other situations, Posidonia, where this relationship completely breaks down. We've learnt that provenance, so where the carbon has come from, and recalcitrance, so recalcitrance is the likelihood that a carbon molecule will succumb to microbial attack. Understanding these properties is very important for understanding the vulnerability of blue carbon. So here we have our 3 blue carbon habitats. You've got the seagrasses, you've got the mangroves, and the salt marshes. They're trapping carbon that's coming in from the water column in the form of plankton. They're grabbing carbon that's coming off the land. And then we can analyse these different forms of carbon using techniques like NMR. We can look at the different parts of the plant material that are left over, plant exudates, which seem to be consumed quite rapidly, up to lignin. We've got terrestrial forms of carbon, which are so recalcitrant. The microbes would burn more energy trying to break it down than they would ever gain in return, so they don't do it. So understanding these different levels of recalcitrance time reflects the residence time they will have within the soils, but what we ultimately end up with is this recalcitrant carbon pool. And interestingly, one of our latest findings, which we haven't yet published, is that a lot of this recalcitrant carbon pool is made up of microbial necromass and residues, junk, left over by microbes that they even struggle to break down. So that's an important part of our carbon stock as well, what the microbes leave behind. We learnt that seagrass carbon is vulnerable to disturbance. I think many of us have experienced the frenemies, so these extreme weather events, hurricanes, oil spills. I was speaking to Mike Duraco last night. He said, I was asking him how things are going. He said, well, seagrass is disappearing widespread again, so now we've got funding again. So, these frenemies, these events that happen which seem to destroy seagrasses have always been a big cash injection for the seagrass community. We'd rather not rely on them. But it's amazing when we do have these events, we realise, well, if we had have had seagrass there, we might have been much better off. Hurricane Katrina, when that went through the Gulf of Mexico, that was certainly the consensus, and that was the response of the Obama government. Let's put back in this green infrastructure. Seagrasses, salt marshes, mangroves, these are a really important form of stabilising our shorelines and buffering against extreme weather events. So this theme of disturbance is something that I've been really obsessed with over the past few years. And I remember we were just about to do a media release on a paper, and a reporter had this moment where he said, wait a minute, so seagrasses sequester a lot of carbon. They've been doing it for thousands of years. There's 20 billion tonnes of this stuff. And it was like this kind of Hollywood moment where he says, what we have here is a seagrass carbon bomb. And I think that was the title of the article. I said, that's about right, actually. We have enormous quantities of carbon bound within seagrass meadows. We lose seagrass meadows. We have lost a lot historically, and in many parts of the world we're still losing the seagrass meadows. And what we have are potentially invisible gases that are being released into the atmosphere and accelerating climate change, going unnoticed. Very different to if you say were to chop down a forest and remove the carbon. It's quite visual. So we've been studying different forms of disturbance. And what I've tried to do is, this is just a snapshot of some of the different forms of disturbance we've studied globally with colleagues. One of the earliest papers to make the case that studying seagrass disturbance and mangrove salt marsh disturbance and its impacts on climate change was from Pendleton et al. Estimated up to 1 billion tonnes of carbon each year is being released as a result of damage, and that results in large amounts of economic damage. So these are some sites we've worked on around the world studying different forms of disturbance, and what I've done is broken it down into 3 main types of disturbance to show you what the outcomes were. So the first one we studied was physical disturbance, going in and just ripping up a seagrass meadow, whether it be through anchor damage or through propeller scarring or just mechanical removal of some form. And here we were studying in Southeast Australia, an area where seagrass had been mechanically removed back in the 1960s. Still hadn't grown back, circular holes about 40 metres wide. It's a Posidonia seagrass meadow which is encroaching and filling in these holes at only about 2 centimetres per year. So we know that the holes have been there for about 50 or so years. And when we cored these environments, this is using a vibrocoring technique, a hell of a lot more work than just banging a piece of PVC, but this is when we want to take deep cores and do paleo reconstructions and spend a lot of money analysing each individual core. And what we found is that in the disturbed areas, the bare sand, there was 72% less carbon in the top metre than in the undisturbed environments. And probably what was most interesting was that decline in carbon, 72% that was lost, represented 1,000 years worth of carbon that had been sequestered, and it's gone in 50 years. So it can go out much quicker than it goes in. And one thing that's still been a mystery for us is that, okay, so even though the carbon disappears when you mechanically damage these meadows, does it just get pushed 50, 100 metres that way and still get bound? Is it recalcitrant enough that it stays and doesn't get converted into CO2? So in this study, we didn't quite answer that, but what we did do was, and apologies for the smallness of this graph, you're just going to have to trust me, that we looked at the microbes using 16S DNA sequencing, and we discovered that in the bare environments, you've got a lot of active aerobic heterotrophs in those environments, and in the undisturbed environments, you have many more of the bacteria that are associated with slow breakdown. They're not that active. So this was an important clue for us that indeed microbial activity and the community composition is quite different in a disturbed environment versus an undisturbed environment. We also looked at the stable isotopes, and the amount of seagrass carbon contributing towards the sediment is diminished in those areas where you've lost seagrass. So the seagrass are no longer contributing carbon, as you might expect. The next form of disturbance we looked at was biological. And at the time when we were thinking about this, Tricia and Atwood and I were talking about some recent examples we'd come across. One of them was from Margarine, who presented at an ISBW, it might have been Thailand or Brazil. And what she showed was that in areas where seagrasses were becoming overabundant, turtles are becoming overabundant, instead of just eating the above ground material, they start to dig down and eat the roots and rhizomes below the surface. And this was destabilizing whole seagrass meadows. And it was possible that this was due to a loss of top-down control, fewer predators, sharks perhaps, in those environments. Now sharks might not eat too many turtles, but they sure as hell scare them and alter their behaviour, the ecology of fear. I mean, the same reason why many humans think twice, especially in Australia, about swimming. So the ecology of fear is a very powerful thing, and we started to explore, well, maybe top predators are playing a really important role in structuring food webs and ultimately influencing carbon. Again, the journos had a field day with this, sharks influence climate change. And then in Cape Cod, we saw in salt marsh environments where we found some great examples where overfishing had reduced the number of large predatory fish in the area. As a result, they'd seen around that same time when it happened, booms of crab populations in the salt marsh, which are just incredibly high densities, burrowing away and reducing the integrity of those salt marsh meadows. And so, this was a perspectives piece. It wasn't a review. There's not a lot of literature to look through. I think there's a lot of work that still needs to be done. But the evidence that we did find was that in areas where you have high predation, this is the sort of creamy blue coloured boxes, you have higher levels of blue carbon. And where in red, this is where there's low levels of predation, where you've lost in a controlled environment, you have lower carbon stocks. And so we proposed theoretically, well, perhaps by manipulating top-down control, reintroducing predators, rewilding, you could influence the amount of carbon in a given seagrass meadow, or a salt marsh, or a mangrove. So you might find an environment where there's not a lot of carbon there. It's some beautiful part of the Caribbean where you just want turtles, good for tourism. So you don't want sharks, you don't care about blue carbon. So that's one option there. At the other end of the spectrum, you might have an area where you really care about predators. And so there are potentially trade-offs we can have with the area in blue shown, where the amount of blue carbon you could have in a typical seagrass meadow. The next and third example are chemical forms of disturbance. So as we started to learn more about the microbes, and one of the things we did here was we took deep cores, this is up to 80 centimetres, and we analysed the number of active microbes in different parts of the core. And this is using flow cytometry. Flow cytometry, when you get high DNA populations, those are the actively dividing bacteria, so they come up as more dense. And then you've got a low DNA population, inactive microbes. And we found that at the top, not surprisingly, where there's a lot of oxygen, a lot of nutrients, great environments for them to be active, we had really high levels of microbes. But by the time you get down to even 10 centimetres, but up to 40 centimetres here, 40 centimetres and beyond, you've only got about 2% of the number of active microbes than what you have at the surface. So we said, well, what if you took sediments from different depths, let's compare deep and to surface, and you then expose them to oxygen? Could you trigger microbes? Could you wake them up? Could you cause them to metabolize that carbon? And the answer was yes. When we took deep and shallow sediments and we exposed them to more oxygen, this is using microsensor technology. Casper was involved, a number of other people here from UTS, Stacey Draven-Tackett, using microsensor technology, looking at oxygen flux and the breakdown of carbon. We did see a significant increase in the breakdown of carbon when we took the sediments and exposed them to oxygen. We see the microbes kicking into gear. Stacey Trevathan-Tackett and Alex Thompson, who are here in the audience, took this one level further, and they picked two scenarios. One is where nutrient runoff into the coastal zone. Another is bioturbation. Bioturbators coming in and pumping oxygen and nutrients down into these deep layers. And some of these callineaster shrimp can burrow down to a metre. You've got maybe 5,000-year-old carbon being exposed to oxygen, nutrients, non-limiting conditions. And what they found was that you can wake up these ancient microbes that are otherwise dormant and cause them to metabolize this carbon. And they did it in a very clever way. They added fresh forms of carbon that they labelled. And then what they did was they looked at what's the basal amount of CO2 release. So there's often some basal level of remineralization, some level of CO2 release. And then they measured the amount of labelled carbon that was emitted and released into the water column. which is this stuff just right here, the dark box here. And then what they found was there was an extra unlabeled component. This was the stuff that was down there deep, that was unlabeled, that was being metabolized in addition to the basal level. So you get a disproportionate increase in breakdown of that ancient carbon. So normally they don't break down too much of it because it's a high energy demand thing, but when you expose them to nutrients or to oxygen, you can trigger them into metabolizing that ancient carbon. So pulling together a lot of this information, the dynamics of blue carbon, the distribution of carbon, all this knowledge we're generating about what we used to have and what we have now. Don't know how I did that jump. We want to move to the next stage, which was, well, how do we better manage our coasts? So these are some of the examples we're exploring internationally. This is what we have now, existing vegetated coastal habitats. We restrict a lot of freshwater flow through dams, weirs, that sort of thing. We restrict tidal flow, which limits seagrasses moving up the shoreline, restricts mangroves moving up the shoreline. We have lower levels of top-down control. As consequence, we have higher levels of bioactivation. But looking to the future, there are, as Rod often puts it, the different management levers we can pull to optimize blue carbon. We can restore hydrology, we can reduce nutrients, we can retreat and move vulnerable areas to sea level rise back up to shore, and ultimately manage for healthier systems, green infrastructure that will respond better to climate change and will help offset emissions. So this is part of an Australian Research Council grant that we'll be exercising for the next 3 years and hope to report out at the next ISPW. In Australia, we're really fortunate that our government has really jumped onto blue carbon, mainly because there's been so many great blue carbon researchers in Australia, and because of CSIRO, we had a large program understanding the distribution and dynamics of blue carbon. And as much as it might seem externally that our governments, our two major parties, Liberal and Labor governments, are fighting about how to deal with climate change, there's one thing they agree on, and that is biosequestration. Our Labor government regards biosequestration as central to any ambitious global effort to meet targets for limiting temperature increase. And then their opposition, the Liberal government, regards biosequestration as the single largest opportunity for CO2 emission reduction in Australia. And then at the last COP meeting in Paris, COP21, our Environment Minister, Greg Hunt, announced that Australia will increase its understanding of and accelerate action on the important role of coastal blue carbon ecosystems in climate change action. And there's two things we've seen happen already since that announcement. The first was that Australia has decided to include blue carbon in its national greenhouse gas inventory, which means we need to understand how blue carbon levels are changing with environmental change. And the other thing is that we've just submitted a report to the federal government, a technical review of blue carbon offset opportunities in Australia. And Carlos brought up too that there's a verified carbon standard brought out by Restore America's Estuaries, and these are, okay, if you do want to do blue carbon offsetting, what might that look like? What are the ways you could do that, and how can you monitor that? So I'd encourage you to look at those reports if you can. So, is seagrass blue carbon just mud? I hope that throughout this talk you've seen that it is, but it's more than that. It's an opportunity, an innovative, I think, mechanism for financing restoration of seagrass meadows, for improving fisheries, for cycling nutrients, and most of all, really, for helping to mitigate climate change. I want to thank all these people that really do the hard work in the group. Several of them are speaking this conference. We've got Brooke, we've got Stacey, and we've also got Alex. Work with a really large number of scientists that have made the science so much better because they've taken a collaborative approach to blue carbon, and you need to because it involves microbiology, omics, geochemistry, so much. So it's been really good in Australia having all these scientists come together and work towards this. And then of course, all the different funding agencies. And I just want to end with two cheeky adverts. We have a PhD scholarship available to study coastal ecosystem services. If anybody's in the market, we'd love to find somebody with a background in social science. So we have a new Mapping Ocean Wealth program with the Nature Conservancy, where we're putting hard numbers on what seagrasses, salt marshes, mangroves do for us in terms of ecosystem services. And if you do want to move to Melbourne, we are the #1 in the world, the most livable city, 5 years in a row, according to The Economist. Now, The Economist is an American magazine. It's not something we just pulled out. So it's the most livable city in the world, apparently. And the second advert is for a new global, the world's largest wetland tea party. It's a terrestrial initiative that we're wanting to take aquatic. The process involves, we will send you, you commit to it, you follow the protocols, it takes a couple of years. We send you tea bags, green tea and rooibos tea. The tea has a very distinct breakdown rate. So you go to a seagrass meadow, you put it out in replication and you throw these tea bags down. And if you've ever done leaf litter decomposition studies, they're a pain in the butt to prepare, right? So you've already got the litter bags prepared, tea bags, right? So you go and you throw these tea bags in. Seagrasses, if you've got salt marshes, mangroves, any other environments, wetlands, you want to put them in terrestrial, that's fine too. We want to be able to partner up with people in this room around the world to start looking at decomposition globally. If you want to be involved in that, please email either Blue Carbon Lab or talk with Stacey Trevathan Tackett here in this audience. She can talk you through that process. We send you the tea, you send the data, and we're going to have one big sort of global paper that we also hope that you'll use that data for your own studies and publish that separately. Now, I wasn't sure if I would finish early, which, have I finished early? I'm not sure. Tell me too. Did you want to do questions or do you want to just...
We can have a quick question.
Okay. And in the background, this is sort of just as a stocking filler, a bit of a seagrass video, which works much better when you've got sound, but it shows some... This is probably the coolest seagrass research I've ever done, involving Macreadie and his team and Paul York. And again, it sounds much better when you've got Top Gun playing, but I don't know how to turn on the volume. Any questions while that plays in the background?
So you said predation is important for blue carbon sequestration, but at least in a lot of the Caribbean, the sea turtle population in a lot of areas is a long way from recovered. There have been some highly published recoveries, but in a lot of other areas, you still really don't see them. How do those seagrass beds compare in terms of blue carbon? Are they the same as if there were a lot of turtles and sharks, or are they some third way?
Yeah, I mean, there were really, in that paper, there are only two examples, one from Bermuda and one from Indonesia, where turtles have become overabundant. We know in many parts of the world there's been a large decline, but we do see in many other systems mesopredator release, trophic downgrading, loss of top predators, and the rise of the next generation of predators. Now, it probably doesn't matter unless they're a herbivore of seagrass meadows, or if they're a bioturbator that just disrupts the sediment geochemistry. So, yeah, I guess what I can say is that, you know, there are only a couple of examples, and in many parts of the world, turtle overabundance is not an issue.
I wonder if you'd explain your difference of measuring the accretion rate in your samples versus the lead 210.
Yeah. So, knowing the stock is good, but it's really only if you lose that stock, and the carbon markets are based on additionality. So, you either have to demonstrate you're going to avoid emissions through loss of carbon, or you're going to increase the amount of carbon gains. And so an important part of that is taking the stock and then working out what was the accumulation rate. Did that top meter of carbon accumulate over, 100 years or 5,000 years? That gives you the carbon accretion rate. And we use lead-210 dating, where we age and develop a chronology for the sediments. But then once you get a certain depth, lead-210 dating doesn't work, so you have to use carbon-14 dating. And the other technique we use are surface elevation tables. We use marker horizons. We use several other techniques just to get an idea of contemporary accretion rates. But lead-210 is what we use most of all, but it's really expensive. It's
not only that, sometimes the signals aren't very great, depending what kind of carbonates you have.
Yep. So if there's been bioturbation or mixing of the sediments, you don't get a decent lead-210 date. Yeah.
Thank you very much, Peter. That was an excellent talk. I was really